In the previous two sessions, we have discussed about the activation of fatty acids and the transport of activated fatty acids into mitochondria. In this third session, we will be talking about oxidation of fatty acids, the beta oxidation of even chain fatty acids. Objectives of this session are: at the end of the session, the learner shall be able to describe steps of beta oxidation, give details of energetics, explain regulation of beta oxidation. Okay, as I said, what is beta oxidation? Beta oxidation, the fatty acid at the beta carbon, oxidation takes place, leaving two carbon molecule. Okay, so that is a successive oxidation of a long chain or medium chain fatty acids. In this session, we'll be discussing about even chain fatty acids. Okay, then what are the sequence of reactions? The first thing is, what are the sequence of reactions? Sequence of reactions in the process of beta oxidation of fatty acids. There are four reactions which are repeatedly that take place. There are four reactions repeatedly that take place, releasing the two carbon fragments. The first one is oxidation. Oxidation. Okay. Then the second one is hydration. Addition of the water molecule. Hydration. Okay. Then the third one is again oxidation. Oxidation. Then the fourth one is thiolytic cleavage. Okay, thiolytic cleavage. So you can say in short, we can say fatty acid oxidation, OART. Oxidation, hydration, again oxidation, and thiolytic cleavage. Okay, let us see how it takes place. Let us go back when fatty acid is not activated. That is a simple fatty acid. I will give an example of fatty acid, then I will show it how it is taking place. This is a fatty acid. This is a fatty acid. We have already studied this fatty acid is activated with the help of the coenzyme. The enzyme is thiokinase, and here there is ATP is converted to the AMP plus PPI in the presence of magnesium 2 plus, leads to formation of fatty acyl coenzyme A. Fatty acyl coenzyme A. Okay, it is called fatty acyl coenzyme A. This all takes place in cytosol. Of course, the enzyme is present at the outer membrane of the mitochondria, the thiokinase. Okay, then the transport is taking place, that is through the carnitine shuttle system. This is carnitine shuttle system. Okay, here, so this fatty acyl coenzyme A enters the mitochondria through the carnitine shuttle system. This is what we call mitochondria. Okay, then again same thing we'll write here. It enters the mitochondria. So it is called fatty acyl coenzyme A. After entering the mitochondria, the four reactions that takes place in sequential manner. The first one is oxidation. Where the coenzyme FAD is converted to FADH2. Okay, this is the first one. Here, the enzyme is acyl CoA dehydrogenase. So this is alpha carbon, this is the beta carbon. The oxidation takes place at the beta carbon. Okay, here the dehydrogenation has taken place. This is called alpha beta. Alpha beta unsaturated acyl coenzyme A. Now the second reaction. What is the second reaction? That is hydration. Here water molecule is added. So here we can say the enzyme is enoyl CoA coenzyme A hydratase. Enoyl coenzyme A hydratase. Then what happens here? Let us see. Hydration is taking place. Okay. So here the hydration is taking place. So this is what we call it beta hydroxy acyl coenzyme A. This is the second reaction. What we call it? Hydration. Then let us come to the third reaction. Then again we say oxidation. Here again the coenzyme NAD plus is converted to NADH plus H plus. Okay, what is that enzyme? Beta hydroxy acyl coenzyme A dehydrogenase. Okay, what happens here? Here dehydrogenation is taking place. Leads to formation of beta keto acyl coenzyme A. Beta keto acyl coenzyme A. Okay, so that is what we call it, beta keto acyl coenzyme A. Beta keto acyl coenzyme A. Okay, then in the next reaction, thiolytic cleavage, the fourth one, thiolytic cleavage. So this is the fourth one. Okay, here coenzyme A is there, enzyme thiolase. What we get here? We get acyl coenzyme A and acetyl coenzyme A. This is acyl coenzyme A. This acyl coenzyme A again going back and participating in the first reaction. It is participating in the first reaction. That is the how repetition is taking place. This acyl coenzyme A, 2 carbon less from original. Okay, so then again another round of the cycle, another acetyl coenzyme A is released. The end product is acetyl coenzyme A, and acyl coenzyme A is again participating in these sequential reactions. Okay, next is energetics of beta oxidation of the fatty acids. Energetics. Okay, let us take palmitoyl coenzyme A, 16 carbon palmitoyl coenzyme A. Draw this one in this way. This is the first, second, third, fourth, fifth, sixth, and seventh. Okay, here you can see. Okay, again I will draw here. Okay, then here it is. Okay, here what we need cycle? FADH2, NADH. Okay. Okay. So like this. What we get is ultimately here there is acetyl coenzyme A, again acetyl coenzyme A, then again acetyl coenzyme A, acetyl coenzyme A, acetyl coenzyme A, acetyl coenzyme A, acetyl coenzyme A. Okay, so there are 7 FADH2, okay, 7 NADH, and 8 acetyl coenzyme A. Here it is 7 into 1.5. Here it is 7 into 2.5. Here, okay, each one 8 into 10. Okay, leads to ATP. So 10.5, 17.5. Okay, so then 80. Then what is the total production? 108. It is total ATP. But what is net gain? Because we are spending 2 ATP spent. 2 high energy phosphate bonds are spent. Okay, then 106. This is net gain. This is net gain. This is about energetics. Now, the third one is regulation. Regulation of beta oxidation of fatty acids. Okay, carnitine, acyl transferase is the regulatory enzyme. This reaction is the rate limiting reaction. Okay, limiting reaction. At this step, the regulation is taking place. So, this enzyme is an allosteric enzyme. Okay, what is the inhibitor? The inhibitor is malonyl coenzyme A. The malonyl coenzyme A is an intermediate of fatty acid synthesis. That means it is very clear. When beta oxidation of fatty acids taking place, fatty acid synthesis will not take place. When fatty acid synthesis takes place, beta oxidation cannot take place. That means synthetic and breakdown reactions cannot take place simultaneously under normal physiological conditions. Okay, let us see in well-fed state why beta oxidation cannot take place. Here, there is increased malonyl CoA, malonyl coenzyme A. So, this one decreases beta oxidation because CAT1, it inhibits the CAT1. Okay, then in starvation, malonyl coenzyme A is decreased. Okay, in that condition, there is increased beta oxidation. That inhibition is not there. So the beta oxidation will take place. So this is about the regulation of the beta oxidation. On the topic of oxidation of fatty acids, in this session, we have discussed about beta oxidation of even chain fatty acids. Let us summarize. Sequence of reactions are oxidation, hydration, oxidation, and thiolytic cleavage. Okay, fatty acids. Suppose take the example of palmitic acid. The palmitic acid, which is activated by the action of thiokinase in the presence of ATP converting into pyrophosphate, coenzyme A is present, leads to fatty acyl coenzyme A. It takes place in cytosol. It enters the mitochondria through the carnitine shuttle system. The fatty acyl coenzyme A undergoes 4 reactions repeatedly. The first reaction is acyl CoA dehydrogenase catalyzes it, that is oxidation. FAD is converted to FADH2, leading to the formation of alpha beta unsaturated acyl coenzyme A. The second reaction, hydration, catalyzed by enoyl CoA hydratase H2O is added, leading to the formation of beta hydroxy acyl coenzyme A. Then the third reaction is again oxidation catalyzed by beta hydroxy acyl coenzyme A dehydrogenase. NAD is converted to NADH, okay, leading to the formation of beta keto acyl coenzyme A. The final reaction is thiolytic cleavage catalyzed by thiolase. Coenzyme A is added, leading to the formation of acetyl coenzyme A, acyl coenzyme A. Acyl coenzyme A is 2 carbons less than the original, that goes again and enters the first reaction. It is repeated again and again. Okay, so this is the how the beta oxidation of fatty acids are taking place. The oxidation at beta carbon is taking place. Okay, energetics. As I said, we have taken the palmitic acid. 7 cycles takes place. This is 1, 2, 3, 4, and 5, and 6, and 7. In each cycle, 1 FADH2 and 1 ADH is produced. 1 acetyl coenzyme A is generated. Okay, then. So at the end of the complete oxidation of palmitic acid, 7 FADH2, 7 NADH, 8 acetyl coenzyme A, leading to the formation of 10.5, 17.5, 80 ATP. Total generation 108 ATP, so 2 ATP are spent. The net gain is 106. Now let us summarize regulation. The rate limiting reaction is carnitine acyl transferase 1. It is an allosteric enzyme. Inhibitor is malonyl coenzyme A. Malonyl coenzyme A is a substrate for the fatty acid synthesis. The substrate of the fatty acid synthesis is inhibiting the beta oxidation of fatty acids. That means when beta oxidation takes place, fatty acid synthesis will not take place. When fatty acid synthesis takes place, beta oxidation does not take place. Okay, let us see the two scenarios: well-fed state and starvation. In well-fed state, so malonyl coenzyme A levels are very high, so beta oxidation will be decreased as carnitine acyl transferase 1 is inhibited. Then the starvation, malonyl CoA levels are low, that inhibition will not be there, so beta oxidation of fatty acids is increased. That is the what the cell demands it. Okay, so with this we have completed the beta oxidation of fatty acids. In the next session we'll discuss about the beta oxidation of odd chain fatty acids. Okay, thank you very much.